ATP synthase is the enzyme that takes part in oxidative phosphorylation. It comes after the electron transport chain. So remember that the electron transport chain pumps H plus into the intermembrane space, which generates an electrochemical gradient. ATP synthase is going to utilize that electrochemical gradient to synthesize ATP. And before we look at the mechanics of how that works, let's take a look at ATP synthase itself. So we can make note over here that ATP synthase has three main components. It has the F1 component, the shaft, and the F0. So the F1, it says it's sticking out on the matrix side. So what does that mean? So if we look at a membrane, and these are our phospholipid heads, and then we also see tails, so these are fatty acid tails. Essentially what we're saying is that F1 is sticking out on the matrix side. So if I denote this side, so this is the matrix side, and this is the intermembrane space. So essentially, when we talk about F1, we are essentially saying that F1 is going to be found sticking out on the matrix side. So we're going to see F0 bound to the membrane and F1's sticking out into the matrix side. So we can see over here, F0 is going to be this part over here that's bound to the membrane, and then F1 is right over here, and it's sticking out onto the matrix side. Now this is very important before we go into detail discussing the bind change model. So F0 is the part that's bound to the membrane, and it is essentially the proton pumper. So remember that we have protons in the intermembrane space, and F0 is going to pump those protons from the intermembrane space to the matrix, and it rotates. So it rotates, and as it rotates, it shifts these protons from the intermembrane space to the matrix. And due to the rotation of F0, we'll see the shaft rotate as well. And when the shaft rotates, it causes a conformational change in F1. As a result, we get the synthesis of ATP. Now, something to go over is that F1 is a hexamer of dimers. Now, what does that mean? This is really important to understand. So a dimer is essentially something that has two components, and a hexamer means 6. So our first dimer is going to look like this. So it's essentially, it comes in pairs. Okay, that's one way to think about it. So we have these pairs, and then this is our pair number 2, and this is our pair number 3. Now in total, we have 1, 2, 3, 4, 5, 6 individual components. But overall, we have 3 pairs. So we have pair number 1, pair number 2, and pair number 3. So we have 3 dimers, or 3 pairs, and overall we have a hexamer. Okay? Now that we understand a little bit about ATP synthase structure, let's talk about the bind change model. So in the bind change model, there are 3 conformations that F1 can have. So remember, F1 is right over here. F1 is the part that is sticking out into the matrix. So when F0 pumps H+, it causes a rotation in F0, and that results in a conformational change in F1. So what does that conformational change look like? So let's say we start off in this conformation, and we pump 3H+ from the I'll denote it as intermembrane space, and those 3H+ get translocated into the matrix. That translocation of H+ is going to cause a conformational change in F1 of ATP synthase. Now there's 3 states in which ATP synthase can exist. It can exist in a tight conformation, an open conformation, and a loose conformation. In a tight conformation, ATP is bound. It is bound to ATP synthase, specifically F1. In the open conformation, ATP is released, and in the loose conformation, ADP and inorganic phosphate bind. It's important to note that all of these things, they're binding on the beta subunit of each dimer. So this is a dimer over here. So the dimers are essentially made up of an alpha subunit and a beta subunit. So we have this first dimer, second dimer, third dimer. We're going to focus on the dimer denoted in orange. So when we move, so right now, if we look at the orange, it's in the loose conformation. If we if we translocate 3 hydrogens from the intermembrane space to the matrix, our shaft is going to rotate, and as our shaft rotates, it causes a conformational change in F1, and we see that we've changed conformation right over here. We went from being loose, and now we're going to become tight. Okay, so ATP synthase, it was loose, and now it becomes tight. Now, if we pump another 3 hydrogens from the intermembrane space into the matrix, so we translocate 3 hydrogens through F0, we see another conformational change occur in the orange side over here. So we started off loose, then it became tight, and now we can see it became open. It became open because we see the release of ATP. So let's go back and look at these again. So we started off in this conformation. This is our starting point. Starting point, if we focus on the orange part, and the orange part is in the loose conformation. We pump 3H plus, it gets converted into the tight conformation. We pump another H plus, 3H plus, we're in the open conformation. If we pump another 3H plus, we end up back where we started in the loose conformation. So the bind change model essentially says that ATP synthase, the F1 component, is just switching from all 3 of these conformations to essentially synthesize ATP. So let's take a closer look at what's happening in each conformation. So when we started off in the loose conformation, ADP and inorganic phosphate just bind. That's it. They just bind. As we switch into the tight conformation right over here, the tight conformation is what is going to be the step in which ATP is going to be synthesized, okay? So ATP is made in the tight conformation. And then in the open conformation, that ATP is released. And that's how ATP is, so this is oxidative phosphorylation. That's how ATP is made using the bind change model. So here we can see that it takes a total of 9 hydrogens, the translocation of 9 hydrogens from intermembrane space to matrix to make 3 ATP. Because remember here we can see we're making 1, 2, 3 ATP, right? Because as the conformation of one dimer is changing, the conformation of the other dimers is changing as well. So this isn't that one dimer is changing at a time. All 3 dimers are changing as a result of every 3 hydrogens pumped. We were just focusing on one specific component, but we have to realize this is happening for all the dimers. So as a result, if we look at, for example, this dimer, we go from tight, then it becomes open, then it becomes loose. If we take a look at the other dimer, we went from being open to tight to loose. Oh, pardon me. We went from being open to loose to tight, right? So all of the dimers are essentially, their conformation is changing. And that is the bind change model of ATP synthase. We utilized 9 ATP and 9 hydrogens to make 3 ATP. And it was F0 that was translocating the protons. The shaft rotates as F0 rotates, and then F1, the conformation of F1 changes as the shaft rotates. And that's how we get ATP after electron transport chain has occurred.